Hi everyone, I am Serife. Today I'm going to talk about the effects of chemotherapy drugs on voltage-gated sodium channels in breast cancer cells. First of all, it would be good to see the relation between ion channels and cancer. And as you can see here, calcium and potassium channels are most well-known types since they involve in self-sufficiency in growth signaling, increasing the proliferative ability of cancer cells through the mitogenic activity. Also, calcium signaling is important for determining the ultimate effect on telomerase activity to provide the limitless replicative potential for cancer cells. Also, calcium signaling can contribute to the formation of new blood vessels in order to stimulate endothelial cells to release and produce some of the angiogenic growth factors to inducing the angiogenesis. It's not working. Yeah. Also, calcium influx, decreasing the level of the calcium permeable channels with activation mechanism other than store depletion can provide or promote cancer cells to evade from apoptosis in order to be resistant to the cell death. In this project, we have focused on to the voltage-gated sodium channels to stay involved in the activating the invasion and metastasis. As you can see here, some of the subtypes of the voltage-gated sodium channels. They are shown before in breast, prostate, and cervical carcinoma types. Also, the presence of sodium current rather than excess of a specific voltage-gated sodium channels protein is crucial for metastasis because in highly metastatic breast cancer cells, especially NAV 1.5 was found to be 1,000-fold overexpressed. Sorry, I'm not working very well. Yeah, my title, where is my title? Really interesting because of the computer. I'm not sure, but it should be to see the roles of the voltage-gated sodium channels in metastasis and invasion in breast cancer. Here is the sum of the latest and previous data from Berkenbury lab. You can see here these are the breast cancer tissue microarray were subdivided into low and high expression of NAV 1.5 in order to understand the metastasis-free survival analysis graph. And according to this graph, as you can see here, we can say NAV 1.5 channel associates with increased metastasis in breast cancer patients. And here is the another previous data from Berkenbury lab. You can see here, these are the bioluminescent image that are control and phenytoin-treated metastatic mice model. Phenytoin is a type of voltage-gated sodium channel blocker, and after 4 weeks implantation, the bioluminescent size were measured from the indicated anatomical site, as you can see here, and according to result after the size were measured on the tumorigenic organs, the tumorigenic liver, lung, and spleen were decreased significantly, as you can see here in this graph. So we can say that the suppression of NAV 1.5 activity with phenytoin reduces tumor growth and metastasis in mouse model. We have questioned, does chemotherapy affect voltage-gated sodium channel in breast cancer cell or not? Chemotherapy is the standard care treatment of adjuvant therapy for triple-negative breast cancer cells, MDMB-231. Adjuvant therapy is coming after the traditional surgical or radiotherapy, or if one of these treatment strategies fails. And chemotherapy includes some of the anthracyclines, alkylating agents, and taxanes. Paclitaxel is commonly used as a chemotherapeutical agent, and it functions as a microtubule inhibitor. I mean that it destroys to the cytoskeletal activity of cancer cells. So according to the neurological effects reported, we have questioned, does the side effects of paclitaxel can be due to ion channels or not? The answer can be yes, because there are so many accumulating evidence that's showing about that paclitaxel can affect the voltage-gated sodium channels activity. And here is, I would like to show some existing data of the effects of the paclitaxel on sodium and on voltage-gated sodium channels. In first graph, you can see here the 25 nanomolar of paclitaxel on NaV 1.7 in neuron cells. As you can see here, the current density of NaV 1.7 is increased significantly as compared to the control after they treated with paclitaxel. And this is the effects of 100 micromolar of paclitaxel on NaV 1.5 current in rat cardiac cells. On the other hand, to the neuron cells, as you can see here, the current density of NaV 1.5 decreased as compared to the control in cardiac cells. But our key question is, does paclitaxel affect NaV 1.5 in breast cancer cell or not? So we have aimed to present further evidence for the functional involvement of voltage-gated sodium channels and especially the NaV 1.5 in chemotherapy drugs, testing in highly metastatic MDAMB231 breast cancer cells by using some of the experimental approaches, including trypan blue assays to detect the cell viability, MTT assay to measure the proliferative ability of cancer cells, and thirdly, wound healing assay to see the effects on cell migration, on cell motility. And we have used whole cell patchogram recording techniques to see the NaV 1.5 function. First of all, we need to find out the non-killing concentration of paclitaxel on MDAMB231 cells. To do this, we have carried out trypan blue assay, and among the various doses of paclitaxel, as you can see here, 500 picomolar of paclitaxel was found to be non-toxic. So we can say this concentration is suitable for our electrophysiology studies. And here is the effects of paclitaxel on cell proliferation. We have carried out MTT assay to do this, and two different doses of paclitaxel treated MDAMB231 cells were incubated under normoxic conditions during the 24, 48, and 72 hours. And as you can see here in this graph, 500 picomolar of paclitaxel inhibits the proliferative ability of MDAMB231 cells over the 72 hours. You can see here. So we can say further supports 500 picomolar is a switchable dose for our electrophysiology. And here is the effects of paclitaxel on cell motility. Firstly, you can see here, scratch area and cell morphology were observed under the microscope during the 8, 08, 12, and 24 hours. Also, we have used mitomycin C in order to block the proliferative ability of cancer cells to see just the effects of paclitaxel on cell migration. So according to result, as you can see here in the cell morphology and in this graph, 500 picomolar of paclitaxel inhibiting the cell migration over the 24 hours. Here is the effect of paclitaxel on sodium current. To do this, firstly, cells were treated with 500 picomolar of paclitaxel during the 24 hours and then depolarized from a holding voltage of minus 120 millivolt. According to result in the first, you can see here the traces of the control and cells after treated with paclitaxel. So sodium currents, NaV 1.5 current traces decreased nearly 50% in MDAMB231 cells. So you can see here in the right side graph, these are the whole peak current density. So after our cells treated with 500 picomolar of paclitaxel, NaV 1.5 peak current decreased significantly. When sodium channels open after depolarization of the cell membrane potential, sodium ions will enter inside the cell, so cell membrane potential will be more depolarized. But in some specific situations, channel gate cannot close, so it allows a persistent current through the channel. So here is the effects of paclitaxel on persistent sodium current. Firstly, here is the persistent traces of control cells and paclitaxel-treated cells. And according to this graph, the persistent current traces decreased after they treated with paclitaxel. And in the right side, again, you can see persistent current density. So we can say 500 picomolar of paclitaxel significantly reduces persistent sodium current by nearly 50% in MDAMB231 cells. So here is the effects of paclitaxel on voltage dependence of activation. Normally, sodium channels have two different gates. One is activation and the other is inactivation. Activation gate is dependent to voltage dependent and inactivation is time dependent. To assess the voltage dependence of inactivation, first these cells were held at minus 120 millivolt and then depolarized to test potential in 10 millivolt steps between the minus 120 millivolt to 30 millivolt. So while current density of NAV 1.5 decreased after the cell treated with paclitaxel, we doesn't see any significant effect on voltage dependence of activation. As you can see here, in the half of the voltage activation and slope, the effect is non-significant. And here is the effect of paclitaxel on voltage dependence of inactivation. Voltage dependence of inactivation have been shown to determine the maximal depolarization. So according to this result, we can say 500 picomolar of paclitaxel does not affect voltage dependence of inactivation in the MD231 cells. As you can see here in this graph, the effect of half of inactivation and the slope graph, the effect is non-significant. So to conclude all the results up to now, our low doses of paclitaxel is non-toxic to MDA-MB231 cells, but inhibits the proliferation and cell migration. Also, this low dose reduces transient and persistent sodium current, but has no effect on voltage dependence of activation and inactivation. Also, paclitaxel may reduce expression of NAV 1.5 at the plasma membrane of metastatic breast cancer cells, even at low doses. This is very important conclusion for us. So we can say this may have a positive impact by suppressing NAV 1.5 mediated metastasis. Is this the end of the story? Not yet, because we need to do further work to understand whether paclitaxel functionally impacts on NAV 1.5 dependent invasive behavior or not. So our project is still ongoing. Special thanks to the Will Birkenbury for supervising and leading to my project and others who contribute to my work. And also I would like to thanks to the Scientific and Technological Research Council of Turkey for awarding me a research grant and Sophion Bioscience for travel grant. Thank you.
Thank you. That was a very clear, very nice presentation. My question is a very small one. Regarding the reduction of the current amplitude, did you check the cell size? Because Taxol is going to change the shape of the cell. Maybe you change the volume. My question is,
Cell morphology, are you asking?
No, the size. Yes, you correct for you show density of sodium currents.
Yeah.
What if you don't correct for density, you see still the difference or not? If you look at the total amplitude, not
they
taking.
Are the same.
It's the same?
Yeah.
So you see the difference?
Yeah.
Okay, that was my question. Thank you.
Okay, maybe I can follow up with the question. Nice talk, by the way. It's interesting that you get the isoform that's found in neonatal tissue for NAV 1.5, and that actually shifts the V-half to positive membrane potentials. And so I'm wondering if, I know you studied the activation properties and activation properties of the channel, but do you think the effect that you see in terms of cancer is due to the actual gaining of the channel into the open state, or could it be just some sort of structural consequence of this channel?
I'm not sure about it, actually, because
because if you look at it, see, if you look at what it would take to depolarize this, to get the cell depolarized, to open up these channels.
Yeah, because decreasing.
Do you think cancer cells ever get to that potential? I'm not sure. I don't know enough about them, but it's just a thought to me. Because I know the isoform is quite 10 or 15 millivolts more positive than the one that would be in a human heart, an adult heart.
Sorry, can you make your question clear a little bit? Because
I'm just wondering, is the effect that you see of NAV 1.5 on proliferation in cancer cells, is it due to the opening of the pore, or is it some other aspect of the protein itself, some structural aspect or something?
Yeah, to understand this, we need to do another experiment inside the cell.
But no one's looked at that.
to understand the expression mechanisms.
Sure.
Yeah.
Sure. Okay.
Thank you.
Thanks for the interesting talk. I just had a question about if you get rid of NAV 1.5 genetically, do you phenocopy or do you lose some of the effects of paclitaxel on the measurements that you're looking at?
We didn't do this, not yet, but according to the previous literature data, there are some of the studies about knock off the NAV 1.5. So by using the siRNA, by using TTX block is the type of voltage-gated sodium channel blocker. So there is no current when they blocked. So so we can make it for paclitaxel-treated cells also. Thank you.
Thanks for an amazing talk. I'm just wondering, in relation to one of the questions that was asked, I was wondering, does cancer cause just upregulation of the channel, or is it making it activate more readily at lower membrane voltage? Does cancer change the activation threshold of the sodium channel on the membrane? And is that what's causing the proliferation, or is it just the upregulation of the channel in the cancer?
Yeah, I understand your question. To understand the upregulation related to the cancer cells, we need to make our experiment under hypoxia conditions. Our laboratory also now studying for hypoxia related signaling mechanisms. So to understand this, we need to do
And when you did the punch clamp and you started, did you test at lower membrane voltages if the channel is activated at lower membrane voltages in cancer cells compared to healthy cells?
No, these are dividing cells, cancer cells. Just depends to the depolarization.
Okay.
So we didn't try any at lower depolarization.
Okay. Thank you so much.
Yeah.